Hey, BBio. So we just wanted to go over a few things here on the Schoology page related to this document, the cell structure and transport. These are just open notes that you guys can type right into and just try to answer those questions. The answers are here, so you can always go back and check your answers. What we want to do is just go over a couple of key points that we feel are pretty important that, or also might be confusing for some people as well when we look at the curriculum. Okay, so first thing is prokaryote versus eukaryote. Please remember, prokaryotes are simple bacterial cells. Okay, they don't have any organelles. They have no nucleus. The DNA is circular. They have free ribosomes, and they have a cell wall. Okay, the cell wall is made up of peptidoglycan. Okay, where eukaryotes, those are all these other living cells. Okay, that are not bacteria, loaded with organelles such as the chloroplast, mitochondria, nucleus, Golgi, linear DNA, right? We have X-shaped chromosomes, okay, when they condense down. The DNA contains histones, okay? They're all those histones that are wrapped around the DNA. They're responsible for regulating gene expression and whatnot, okay? So major differences between the two. Eukaryotes are larger, okay? Prokaryotes are very small, okay? On this, we just have a list of the organelles. And again, you should know the name of the organelle and their function and what they do. Okay, so please make sure you guys are good with that. The big ones are these: ribosome, ER, Golgi, mitochondria, and of course chloroplasts. Okay, big time with the chloroplasts and the mitochondria because they both have their own DNA. They are examples of the endosymbiotic theory. Okay, as we scroll through, here's our endosymbiotic theory. Real important, right? All eukaryotic cells evolved from bacterial cells that eventually combined and grouped together and lived together, and they became autonomous. I want to flip real quick to a one of my slideshows here that deals with endosymbionts. Here we go. Okay, let's make this bigger. So the first eukaryotic cells, oof, let me get this to come back up. There we go. There we go. Appeared about 1.5 billion years ago. Again, endosymbiotic theory suggests that energy-producing bacteria came to resolve within larger bacteria. Okay, they have separate DNA. They have double membranes. Okay, this is important because the double membrane illustrates that inner and outer membrane that we see in some bacterial cells. Okay, what this looked like is something like this, where you started off with these cells, okay, that were separate and eventually became incorporated into this non-photosynthetic eukaryote, and they became eukaryotic cells. Okay, better picture here shows, again, you start off with this cell that has just this loose DNA in it. Okay, at some point, perhaps the nucleus was an ancestral cell, and that became involved in this. The wrong thing to get rid of. And then eventually with that cell, it started to pick up all of these different structures along the way, which were the result of these different types of bacteria getting in there. Okay, the remnants left behind, the evidence left behind that shows that these were once free living, double membranes, DNA, separate ribosomes, very similar in terms of what bacterial cells look like. And the DNA, it's circular, right? So they resemble bacteria as well. Okay, so that's the endosymbiotic theory there. Just wanted to touch base with that. Let's jump back. Cell membranes, okay, they're selectively permeable because of the structure. Okay, the phospholipid heads are on the outside, tails are on the inside. Real important when it comes to the movement of materials across the membrane. Okay, so let's jump to that real quick. Okay, and we can see right here, phospholipids. Okay, give this a chance, make this zoom in. Okay, phospholipid bilayer cross section, right? So what we've got here is we've got a lipid bilayer setup. These are your hydrophilic heads, polar, attracted to the water. Non-polar tails, one is always saturated, the other one is unsaturated. That creates a bend, which means more flexibility for the cell membrane. Okay, and these membranes, these phospholipids are always moving around. So you're always going to get little openings, little holes in there, and that's where things like oxygen and nitrogen and CO2 can easily pass through. I'll show you here on this picture. You can kind of see that there are small little openings here. Small uncharged molecules can pass through those openings. Okay, and the key thing is here is that by setting this up this way, we can create two separate environments, okay? And again, both of these environments are watery. They both contain water. They're aqueous. We have the extracellular fluid. This is the outside component of the cell. This is on the outer surface of the cell. And then we have the cytosol on the inside, okay? Ions can't pass through. Even though they're tiny, an ion can't pass through because they get hydrated, right? They attract water, makes them too large, and they have too much of a charge. And so they can't fit through these openings. Even if they could, they get repelled because this internal component of the membrane is hydrophobic, okay? And it's nonpolar. It's going to repel material out of the cell, okay? So it prevents things from moving in, all right? So I wanted to show you that. Again, that's a close-up of our phospholipid. There's the polar head, non-polar tails. Amphipathic is the term. And again, by having that bend, that creates that flexibility and the permeability so that things can get through, okay? So the semi-permeable nature of the membrane is the result of these tails having this bend in them, which creates this little bit of space, which permits certain things, but not everything, to pass through the membrane, okay? We zip out of this for a minute, okay? Transport, real important, okay? So this diagram here shows you the four types of transport we talk about, or really the three. We have plain old simple diffusion, right? And in diffusion, we're just simply going from high to low, okay, down the concentration gradient. This is also diffusion, but now this is facilitated. So facilitated diffusion can involve either a channel protein, that's the proper size, which allows molecules to pass through, or it can involve carrier proteins that physically bind to the molecule. They're a little more specific, and they can pull it across. Things like a glucose transporter, for example, okay? And then active transport is where we're moving things against their gradient. Often we see this coupled, okay? We see that sodium gets pumped out and potassium gets pumped in, okay? And they're going against their gradient. So they're going against the concentration gradient. And actually, gradients don't even matter with active transport. Active transport is moving these molecules regardless of the concentration, but it needs to use energy. Most of the time, the energy is in the form of ATP. And what ATP does is it adds a phosphate to this transport protein and changes its shape so that it can become activated. Okay, when you add a phosphate to a protein, the protein changes conformational form, and that permits molecules to move in and out. Okay, now ATP isn't the only thing that can be used. We can also utilize things like other ions, like hydrogen ion gradients or calcium ions. Okay, and what calcium ions and hydrogen ions do is the same thing that phosphate does. They bind to the protein, change its shape, and allow it to move things against the gradient. Okay. The famous hypertonic hypotonic situation. Okay. If you place a cell in a hypotonic solution, that means that the solution outside of it has a lower concentration of dissolved substances. Water is going to rush into the cell. So if the cell is placed in a hypotonic solution, it's going to explode. Hypo, low, the cell will blow. If you take the same cell and put it into a hypertonic solution, that means it's now placed in a salty solution or a solution with a higher concentration, the water is going to leave. Okay, hyper high, the cell will dry. Okay, this is referring to the solution. Now, this cell that's in a hypertonic solution, the interior of the cell has a lower concentration of solute compared to the outside. So the cell is hypotonic relative to the outside of the cell. If you look at a cell that's in a hypotonic solution, that means that the interior of the cell is hypertonic compared to the outside of the cell. Okay? Iso means the same. So an isotonic solution, water is moving in and out equally. It's in equilibrium, and so the cell does not change. Okay? Now for plant cells, when the cell plumps up, it's called turgid, right? And we call that turgor pressure. And that's what allows plants to stand upright. So when plants get a lot of water, their cells fill with the water and it makes them turgid. And it's that central vacuole inside the cell pushing up against the cell wall, which allows the plant to stand upright. If you take plants and expose them to salt water, they shrivel up and they become what's known as plasmolyzed. And the cell walls don't have that same pressure inside of them, and they become limp and they fall over. Okay, and so that's not good for the plant. Plant's not happy at all. Okay, we don't need nerve action potential or any of this because none of this is going to be there on the exam. Origins of cells, you should just have the idea and the understanding that there is evidence that shows that a lot of the building blocks of life all formed early on, and that all life forms share a common genetic code. That's an important concept, and that we use the same processes like diffusion, osmosis, active transport. If you look at cell respiration in a bacteria, it's pretty much the same as it is in a eukaryotic cell. Okay. There's also a lot of many conserved core processes, right? DNA and RNA, they're the same. Replication, translation, transcription all occur the same. We even have evidence that shows relatedness by looking at things like the cytoskeleton in all eukaryotes. Organelles are the same, okay? And the way chromosomes are set up. So that's all very important stuff when we're looking at how plants and animals are set up, okay? Just a few more things I want to take a look at from the notes that we took in class. Let me just scroll through. Facilitated diffusion, real important concept I want to get to with you guys. Okay, so key thing with facilitated diffusion is it's utilizing membrane-bound proteins without energy. That's an important concept, guys. No energy gets used, so we're moving materials from high to low concentration and then moving down the concentration gradient. Real important. Okay, it's going from high to low, no energy, down the gradient. And you have two options. You can either use an ion channel, like a hydrogen ion channel or a potassium ion channel, or we can use carrier molecules, which are very specific. Okay, and they're going to move molecules specifically from high to low concentration. Okay, examples of these carriers include things like the sodium-potassium pump. Okay, but that's going to use energy. Okay, and so that's using energy is active transport. Okay, and so here's an example here of active transport. Okay, ATP is going to bind to the protein, change its shape, and we're going to move the molecule from low to high. Okay, against the gradient without energy. Okay, the energy from the ATP, from that phosphate. Okay, we're adding a phosphate molecule that is going to enable this protein to change shape so we can move things across the membrane. Okay, real critical. Okay. Without that energy, we can't move things across the membrane and transport stops. Okay, so we really see this when we want to generate ion gradients. Okay, now another interesting example of this is in the mitochondria and the chloroplasts. Remember we talked about the electron transport chain, and we had those cytochromes which were moving electrons along the membrane. Those pump hydrogen ions from low to high concentration. Okay, so that's an example of active transport. But what it's doing is instead of using energy from ATP, it's using the charge energy of the electrons. Okay, as the electrons flow through the membrane, we're using the charge energy to trigger the movement of those ions across the membrane. Let me show you. I'm going to open up one of those. So give me a minute. We're live. Let's go to cell respiration. This looks good. Okay. So these are all those wonderful, lovely class notes that we took. I just want to go right to the diagram here. Here we go. Okay. So here, this arrow right here represents the movement of the electron. Okay. As electrons move through these proteins, they change their shape and allow them to pump hydrogen ions across the membrane against their gradient. That's active transport because we're using the energy from the electron to alter the structure of the proteins to pump them across. Okay, and the same thing happens in photosynthesis. The electrons move through the photosystems, and as they do that, it causes them to pump more hydrogen ions across the membrane. And that establishes a gradient that can be used later on to make ATP. Okay, we'll get into chemiosmosis, but this is facilitated diffusion. This is going from high to low concentration down the gradient. Okay, and as it goes down the gradient, it's making ATP. Kind of interesting. Okay, but the flow of hydrogens from high to low, that is facilitated diffusion. Okay, and again, we're not using energy for that. All right, I think that is everything I wanted to address. But since I have this open, let's take a look. I think you guys should be pretty good with endocytosis and exocytosis. Aquaporins, again, facilitated diffusion. This is moving water molecules through the membrane. Water molecules move through the membrane utilizing aquaporins. Okay, that's a water channel. It's a pore. That's just the right size and charge to permit water molecules to pass through the membrane. Okay, and I think that's pretty much everything that we wanted to get into. So we're good. All right. Again, questions, please fire them away. We're here for you guys. We want to help. Okay. Good luck with everything. Have a great week. See you soon. Bye.